I'd like to start by thanking the organizers for this invitation, and in particular Duncan, because he has been very persistent, and this is the third invitation, so 3 times is a charm. I wasn't able to make the previous 2. So this is, I'm going to talk about, we're doing a lot of things in the realm of L-type calcium channels, which are primarily postsynaptic, they're signaling, and neuropsychiatric disorders. I'm a molecular biologist, molecular neuroscientist, so I'm really interested in signaling and molecular mechanisms. And what I'm going to talk about, I decided to talk about, this is largely unpublished data that I'm really excited about, which taps into learning and memory that happens in addiction. And I will talk about CACNA1C, which codes for the CAV1.2 L-type calcium channels. So memories. Most neuroscientists, have been spending a lot of time trying to preserve good memories, such as in disorders like Alzheimer's or in normal aging. But those of us who are interested in the biology of conditions such as addiction or trauma or fear-related conditions, we actually have to tackle bad memories or unwanted memories, and how do we, these are extremely pathological, that result in these conditions. And how do we fix such conditions? People have thought about erasing these unwanted memories, but as you can imagine, that this is rather challenging to do, given it's hard to target selective memories. So there's another form of learning to forget these unwanted memories that many of us have been thinking about, especially much more done with fearful memories. And what we have been doing, and what I'm going to talk to you about, is learning to forget memories related to cocaine addiction. Learning to forget is an active process, and it involves mechanisms such as new transcription and translation. And as I said, I'm going to share some of our data focused on the CAV1.2 channel and its interaction. For the longest time, I've been studying how these CAV1.2 L-type calcium channels interact with 2 major neurotransmitters, dopamine and the dopamine D1 receptor and glutamate receptors postsynaptically. And too bad Dick Chen is not here, because a lot of work, when I started my work as a postdoc in Boston, I built, a lot of the work was built on wonderful work done in Dick's lab and also my Greenberg's lab. Okay, so within the context of addiction is extremely complicated, and treating addiction is definitely a challenge. We've been focusing on cocaine because there are no existing pharmacotherapies for cocaine addiction. Opioids are a problem, but there are medications. So within the rather complex framework and circuits of the brain, what I'm going to share with you today is particularly learning and memory that takes place, a lot of the mechanisms take place in the brain region, the hippocampus, which has been shown for regular learning and memory. And in particular with addiction, the hippocampus is really recruited when dependent individuals relapse. And this happens due to the strong memories that are drug context association, drug cue association, such as what's shown here. And this is one of the reasons that individuals relapse to drugs of abuse, such as cocaine. And how do we study this in mice? So I'm going to show you, we use a lot of different mouse models, and the work that I'm going to share in the first part was really spearheaded by Caitlin Bergdorf. She graduated just a little before COVID. So I'll walk you through the behavior that we use. It's called cocaine conditioned place preference. It's a simple but a really useful model to study drug-associated memories. So it involves a 3-chamber, it involves 3 chambers, like shown here. On day 1, so this goes over multiple days. On day 1, the animal is put in the center and allowed to freely move across all 3 chambers. On day 2 to 4, the animal is given a shot of cocaine placed on one side of the chamber in the morning, and in the afternoon, the animal is given a shot of saline and placed on the opposite side. And this happens for 3 days, and animals on the 5th day, this is the protocol we use in the lab, on the 5th day are allowed to freely move, and majority of the animals show a preference for cocaine. They love, majority of the animals love cocaine. It's extremely reinforcing. And they spend significantly more time on the cocaine-paired side than the saline-paired side. Now, this is what we refer to as the original cocaine memory. Then the animals can be run through the chamber over days, and eventually what happens, this is in the absence of the drug, the animal learns to no longer associate the cocaine-paired side with cocaine, and will spend less and less time, as shown here, over days. And what we refer to this as the extinction memory, or this is extinction learning. And this is a new form of learning. And we know that the original memory is still there. And this is what the reason for relapse in patients is that you can stress, in humans, or you can give exposure to cocaine. You can do the same thing in mice, and either stress or a shot of cocaine, actually animals relapse. So, and this is believed that it's reactivation of the original memory. So I'm going to show you, we've been focused on, can we actually teach animals to extinguish this cocaine memory? So one of the first discoveries that Caitlin made was that when she was running the animals, the L-type calcium channels have been linked to cellular models of memory, such as long-term potentiation. What she actually found was that there was no change in either the mRNA or protein of CAV1.2 in the original memory, that in this behavioral model, whereas when animals went through extinction learning, there were significantly higher amounts of both mRNA and protein in the hippocampus brain region, and there was no change in other brain reward regions that she examined. So this was kind of exciting, and it was showed us that this is an activity-dependent process, that we were seeing this enhanced CB1.2. And then I don't have time to show you a series of experiments we did. We created tons and tons of genetic animal models, and various different techniques. And what we have found, this was a very interesting discovery, is that within the hippocampus, there are various subregions. The dentate gyrus, this is, we love the rainbow, shown here. What's shown down here is that we found that the CAV1.2 was actually really important in a subset of cells within the dentate gyrus that express dopamine D1 receptors. So what you're seeing here, all the green cells express D1 receptors, telling us that there was something very special about this subset of cells that have CAV1.2. They have D1 receptors. They are excitatory. These are granule cells. They're excitatory. And perhaps they require both glutamate and dopamine for this extinction learning process, different than neighboring cells that receive only glutamate and do not express D1 receptors. So being a molecular, before that, so we have also, we're very interested, are these cells actually recruited, the subset of cells, during this condition-placed preference? So we have been using, this is showing fibrophotometry, where we are able to look at neural activity during behavior. Rob Fecho in the lab was greatly involved in bringing this technique and doing a lot of coding. And what I'm going to show you, and it's really cool. I think that this is methods such as these are really going to bring us closer to answering the question about what are mice really thinking. And what we found is that this is kind of a summary where what we did in order to selectively look at these D1 positive cells, we expressed GCAMP in the D1 cells. And as we had predicted, during extinction learning, there was an increase in neural activity of these cells following extinction learning, but not really during cocaine memory. So these cells are recruited primarily during this learning. Whereas the same, when we, in our CAV 1.2 knockouts within the cell type, we saw no increase in activity. So this kind of gave us a functional readout that these cells are indeed recruited, and CAV 1.2 is important. We're doing this in parallel with a lot of slice physiology that is currently ongoing. So being a molecular lab, I'm summarizing again lots of molecular experiments, but what we have found within spines of these, within the dentate gyrus of these D1 receptor cells, we find that extinction learning requires an increase in CAV 1.2. We have done postsynaptic fractions. It's amazing how much CAV 1.2 is there in the postsynaptic density. And during this learning, the D1 receptors activated pKa is really important for phosphorylating AMPA receptors, the GLUA1 homomers at serine 845, whereas the CAV 1.2, which is important for inserting these calcium permeable, so these are calcium permeable AMPA receptors at the synapse. The CAV 1.2 is actually really important for CAM kinase 2 activation of the CAMK2 site, serine 831, which we believe will increase conductance, again, slice physiology being done. And this process is essential for this learning. Everything I'm showing you here, we have done the causal experiment thanks to colleagues who have given us a lot of tools and tools developed in my lab. So currently, what are we doing? We're really interested in, what is the crosstalk between D1 receptors and CAV 1.2? These are G-coupled receptors. Others have looked at beta adrenergic receptors and CAV 1.2. Is this also happening with D1? So stay tuned for that. Now, I'd like to move to my title. So learning to forget. So could the CACNA1C underlie individual differences to addiction? So we all know that not everyone who uses cocaine becomes dependent. And there is a susceptible population. Here's an example of celebrities that we all know, that who have actually extremely creative, bright, intelligent individuals who have succumbed to cocaine. They're all cocaine overdose victims shown in the slide. Whereas there are always people like, I'm fascinated by Robert Downey Jr., and because he belongs to me, he belongs to the resilient population, and he's brilliant, as Iron Man and Sherlock Holmes. Is what's really driving, what's predisposing the susceptible from the resilient? Another discovery that Caitlin made, and I'm going to, in this last few minutes, going to talk about a new exciting, direction that this project has taken that taps into epigenetics. So Caitlin started this project. This is now being done by Madeline Baker, who's a neuroscience graduate student, with close collaboration with my colleague Miklos Toth, who is a brilliant epigeneticist. So this whole project, so our whole, I said that we have a huge question is what predisposes certain individuals. Of the many factors that probably contribute to drug dependence, genetics is something that many of us have been interested in. Now, it turns out that the CACNA1C gene that many of you know is one of the top candidate risk genes for multiple different neuropsychiatric disorders, and recently also linked to alcohol dependence and lifetime cocaine dependence. I don't have time to talk about this, but there's a lot of comorbidity between these neuropsychiatric disorders and drug use and abuse. But what's interesting is that many of these SNPs are within the intron 3. This is a huge gene, but many of the SNPs, interestingly, are in this intronic region. And it's been a challenge to figure out what is the actual functional consequence of these intronic genes. One suggestion that has been made beautifully and has been shown using schizophrenia-linked single nucleotide polymorphisms, SNPs, is that these SNPs actually dysregulate something called chromatin looping at the genomic level, and disrupts chromatin looping, thus decreasing CACNA1C. So kind of loss of function or lower CACNA1C could be predisposing to schizophrenia and other neuropsychiatric disorders. So keep this in mind as I go through. I'm going to go back to the mouse data. So one of the discoveries Caitlin made was that when she was running just regular C57 mice through the CPP and extinction, she found that these are animals that, where they had the same amount of this original memory, but when they were run through extinction, there were always these few animals that would just not extinguish. And for the trainees in the group, if your experiments are technically sound, don't ignore outliers. And so Caitlin got really interested in, why aren't these animals not failing to extinguish? And what she found through a series of experiments, that one of the first things was that in the dorsal dentate gyre, in the hippocampus of these mice that fail to extinguish, they actually cannot upregulate CAV1.2. So neither CACNA1C mRNA or the protein. So the black bars compared to the animals that are able to learn the extinction, these blue bars. And then all the signaling downstream, she found that none of these, the CAM kinase and the the AMPA receptors and also CREB was not upregulated in these animals that failed to extinguish. So suggesting that the CAV1.2 was really necessary for this extinction learning. So what could be possible mechanisms? We turned to epigenetics. And within all the different, various ways that DNA and histones are modified, we've focused on DNA methylation because of this collaboration we had with Miklos Todd's lab that has been studying DNA methylation and learning and memory. And in a very simplified way of what methylation does, methylation happens at CPG sites on DNA, and if a gene is methylated, it tends to suppress gene expression, whereas in an unmethylated state, it will drive increase in gene expression. So we ran a whole genome-wide DNA methylation study, and basically we were looking at differences in methylation between animals that were able to learn to extinguish versus those that were not, failed to extinguish. And what was very, very satisfying was this was we went and blinded, but when we did this ingenuity assay, our favorite molecules showed up. CAV1.2 was one of the top, CACNA1C was one of the top targets, and many of the other glutamate receptors, CREB, CAMK2, ERK, these are all genes that we had in our candidate gene approach had focused on this. We were really satisfied to see that DNA methylation was altered in many of these genes. And when we focused, my lab being really interested in the CACNA1C, we actually found, when we compared the CACNA1C between animals that learned to extinguish versus those that failed, there's this one region within intron 3 of the mouse. So you recall that intron 3 appears to be sensitive in the human gene. There are 6 CPG sites, and what's really interesting, Diane, you'll find this interesting, is there's a CTCF binding site right within this methylation. And Diane has a beautiful paper on CTCF and splicing of the CAV2.2. It's really interesting that there is a CTCF that does many things, but it's also a methylation sensitive chromatin regulator. And so we have so CTCF is really important for you. One of its roles is in this chromatin looping. And the hypothesis that we have now we are working on is that animals that are able to successfully extinguish is they have this region of the CACNA1C gene is unmethylated, allows CTCF to bind, increasing CACNA1C mRNA and protein, whereas animals that fail to extinguish are hypermethylated in this region and are not able to increase CAV1.2. I was going to say something, but I've forgotten. So going ahead is I just want to show you some data, which is these are the 6 CPG sites. And when you look at methylation levels at each one of them, this orange line is the methylation status of animals that extinguish. And each one of his dots, you can see that they're pretty much unmethylated. Whereas animals, these are just saline treated animals or animals that fail to extinguish have much more higher methylation at these CPG sites. So we have ended up going down this. We have data that suggests that methylation could really be driving these differences. And now we have, Madeline is very busy doing, she has cloned this region. We are very interested. She's doing a lot of cell line work. And then we'll also move it to the mouse. And there are lots of really cool tools. We're looking at other histones and basically how this region is regulated in the mouse. And the dream is to use technologies such as CRISPR to actually go into the mouse and be able to fix it. So as my last slide, I'll just go back to this. This is what has been shown in the human brain using human brain tissue, that there's something special about this intronic side, the ENCODE database that is used to identify domains suggests that what we have found is a silencer element. But perhaps a combination of DNA methylation, single nucleotide polymorphisms, these interactions happening within introns could actually dictate some of the pathology that's seen in neuropsychiatric disorders. So we have lots to do. I'm really excited about, as a molecular biologist, I'm really excited about, this is what I worked on as a graduate student when I was at Purdue, really thinking about transcription at the genomic level. So with that, I will end by thanking It Takes a Village. I've been really fortunate to have wonderful graduate students, wonderful collaborators, and thanks to the funding. Happy to take questions. Thank you.
Hi, thank you. It was super interesting. So I have few questions. The first one is, so you studied dorsal hippocampus, and there are recent data showing that actually ventral hippocampus also has a lot of impact and in the dopamine signaling. Have you ever looked at it?
Yes, we have looked at it from the CAV1.2, like knocking it out and see in the ventral hippocampus. We really think that the ventral hippocampus is driving the reward-related aspects of cocaine, whereas and other addiction. So we have data showing that other addiction-related comorbidities, such as anxiety and depression, is driven by the ventral hippocampus, but it really, Caitlin went and just knocked out the calcium channels in all different regions. The dorsal hippocampus appears to be very specific to learning and the memory of context, whereas the ventral hippocampus receives dopamine from the ventral tegmental area. So we think that these are separable circuits driving different conditions.
So the second one is, you have done calcium imaging, but have you ever measured like dopamine with delight or GrabDA and see also if there was CAV1.2 dependence?
Yes. So we are doing that right now, and we're using in the hippocampus. There's very little dopamine in the hippocampus, but we've been very lucky with the high affinity GRAB DA sensor versus D-Light has been working beautifully in the dentate gyrus. So we are able to, it is too early to show it here, but we see differences in dopamine levels in animals that extinguish versus don't extinguish. So we are doing all those, and they're now Cree-dependent grabs that are being developed. And by the way, the person, going back to Dick Chen, the person who has developed Grab DA was a postdoc in Dick Chen's lab. He's now in Japan.
Great. Last question, and then I will pass the mic. Do you plan on doing attack sequencing?
Yes. So Madeline is, so there's so many experiments we want to do that is on the list of doing that. Right now, we're trying to correlate the methylation with just doing RNA-seq, but eventually moving, and then there's so many databases trying to see, what histone marks are there and so on. But yes, that's the direction we're going. Thanks for the questions.
Hi, really interesting. I've got a question regarding why the mice might have different methylation states.
Yes.
Have you looked at the social status of the mice? Are these group housed? Are they male? Are some of the mice getting bullied and undergoing more stress than the other mice, and that's why they are reactivated, maybe?
Yeah. Great question. So we are going to be doing, these are all in male mice. We have done only preliminary work in female mice, but we're going to do that. So I don't have the answer for that. Great question about what's driving this hypermethylated. So most of our animals are not fighting, and we have found that animals that actually get stressed or bullied, they actually love to have higher cocaine preference, as one would expect, stress times addiction. But we don't know. We don't know whether it could be higher DNA just basally, whether they're higher DNA methylases or decreased DNA demethylases. All questions that we're asking. So we don't know.
Hi, Anjali. I have a question to you. It's a great talk. So have you ever considered using LTAP calcium channel blockers to treat the mice and see if they get rid of the cocaine addiction?
So the LTAP calcium channel blockers, it has no effect on the original cocaine memory. That's the first thing that I actually did when I started my lab here at Weill Cornell. The activator BK, if you give that during extinction, it does expedite extinction. And however, BK unfortunately is, it's not the greatest drug to give IP. So, but we did give it to the brain, in the brain. But we have a collaboration at Yale where we're doing rat self-administration, which is the gold standard to study addiction. And following extended withdrawal periods, we've done experiments with this ratapine and nifedipine, it does attenuate what we call, cocaine seeking. So, and has no effect on sucrose seeking, which is always the control, because you don't really want to dampen natural rewards. So, at least it appears that, and we are doing more studies on that. Yep. Yes, Diane.
Very quickly, so if you look in the ChIP-seq database, do you see for the site that is predicting TTCF9, and do you see differential peaks across different cell lines? Because that's not necessarily demonstrating, cause effect, but it does suggest dynamic regulation. Do you see that?
So I won't be able to remember the exact data, but Madeline actually has done that, and she has looked at, so it does look like we should be able to get some insight at what's happening within that region. And so we are looking at the databases, and it does appear that, that is a region that there is a lot of histone modifications within that region. Yep. Thank you.